Welcome in my poster presentation. The title of my poster is Affinity Measuration of the Structure Guided Single Chain Variable Fragment Antibody Directed to FUVC Epitope of Dengue Envelope Protein. Before going in details, I am discussing some general of dengue virus infections. Dengue virus is highly prevalent in 128 tropical and subtropical countries. Approximately 390 million people are getting infected in each year throughout the world. However, there is no effective vaccines and treatment available yet. The major challenges of dengue treatment is antibody-dependent enhancement effects. The FC receptor of cross-reactive dengue antibody generated during the secondary infections with alternative serotypes plays the key role in antibody-dependent enhancement effects. That's why the developed vaccines and other alternative immunotherapies are not effective and sometimes they are counter-reactive. Therefore, we hypothesized that HCV antibody might be effective against the progressions of dengue virus infections. Accordingly, we have designed an HCV antibody from an dengue anti-fusion loop E53 antibody whose PDB ID is 3IXY. The BH and BL domain sequence of that antibody is joined with the repeated units of lysine 4 serine 3 linker. Initially, we have a homology model of this HCV antibody and docked with the dengue envelope domain to protein where the BC and fusion loop is located by using the Discovery Studio software. From this, we can notice that both this protein is getting interacted. The interaction interface of this dock process were visualized with using the EPI team plot, where we can notice that 5 out of 6 CDRs of HCV protein is getting interacted with the dengue envelope domain to protein. Further structure-based sequence analysis revealed the 3 out of 5 SAP antibody CDRs are getting interacted with the targeted BC and fusion loop, and certain residues of BC and fusion loops are getting interacted with these CDRs. And the multiple sequence alignment also revealed that the BC loop and the fusion loop sequence are highly conserved throughout the dengue serotypes. Based on this sequence analysis and using the virtual platform Discovery Studio JetDoc program, we have created one virtual antibody mutants, HCP mutants libraries where the interactive CDR residues of HCP proteins were mutated with alternative 9 side chain containing amino acids and made a short list of top 10 single mutants, for double mutants and for triple mutants based on the docking Z-track score and the Z docking scores. Initially, we have created top 10 single mutants by site-directed mutagenesis and expressed and purified that proteins by size fluorescent chromatography. The purified proteins are visualized in section 6 by SDS space, where we can notice that all the protein single mutant proteins are purified properly. The purified proteins further utilized for binding studies using the SPR platform, where the FUVC recombinant proteins were immobilized on the SPR sensorgram, and HCP wild type and mutant proteins were applied as the analytes with the different concentrations, and from the association and the association kinetics of his mutant proteins were calculated KD value by using the autolab split evaluation software, and graphically shown in section 8, where we can notice that except the mutants 4 and mutant 10, all of the mutants are decreasing the KD value compared to its wild types. That means all are increasing the affinity except these are two proteins. The single mutants KD, based on this, we have visualized graphically in section 9, where we can displayed the green color zone as the interactive zone of HCP, where the red color is residues are negatively regulating mutants, and the blue color is showing the positively regulating mutants. Based on these single mutants, we have combined with each other for study synergistic effect and created double mutants, and also studied their SPR binding kinetics. However, the binding kinetics of double mutants haven't shown here. However, based on the SPR double mutants results, we have created uh, for combinatorial triple mutants, and similarly undergone for SPR binding kinetics, SPR sensorgram of triple HCP triple mutants from 15 to 18 were shown in section 10. Similarly, for further validations and reproduce the results, we have used the BLI bioeffectometry platform, and the sensorgraph retrieved by this BLI experiment were shown in section 11. The KD value retrieved by SPR binding kinetics and the BLI kinetics were used for calculations, mean and standard deviations. And with the mean and standard deviations of KD values retrieved from BLI and SPR kinetics were plotted as a landscape with a 3D plot where the x-axis is showing the triple mutants, y-axis showing the KD value changes, and z-axis is showing the ability fold change. From this plot, we can notice that mutant 15, who is increasing the affinity around by 100-fold changes, whereas mutant 16 and 15 also increases affinity in a moderate level KD fold change. Whereas mutant 18 is increases affinity in lower KD fold change. Further sequence analysis revealed that mutant 18 is carrying one negatively regulating mutant 16 in 230 tryptophan. That's why so far it changes a lesser amount of affinity. Otherwise, the mutant 15, 16, and 17 have higher KD fold change affinity. That means they have some therapeutic potential. However, before going that clinical studies, it needs further validations with in vivo mouse model studies. This is all of my results. Thank you all. We are also thankful to South Asian University for financial support and providing research facilities. We are also thankful to Jawaharlal University and Regional Center for Biotechnology for providing SPR and PLI facilities. I am also thankful to my supervisor for keen guidance and providing financial support. Thank you all.